Namaste students, I'm Mr. Vidya. I'm JV'n's Dr. Neeraj Dholia, Faculty of Agriculture and Veterinary Science, Jayoti Vidhyapeeth, Women's University, Jaipur. So today we are going to discuss the topic, the mechanism of fertilization and activation of ovum. So this subject is under this, and this session is under the subject developmental biology. So let's start. So as you know that this fertilization is the characteristic feature of the sexual reproduction, and it is defined as the fusion of the male and female gametes lead to the formation of the zygote. So the process in the mechanism have the four different processes, or we can say that this mechanism involves the four processes. One is the production of or preparation of the male and female gametes, these sperm and the eggs. The second is the recognition and binding of the gametes, then fusion of sperm and egg, and then fourth is the activation of the fertilized egg or the zygote. So let's start one by one. So the first step, that is the production and preparation of the sperm and egg. We have already taken the individual lectures for the production of sperm and eggs, the spermatogenesis and the oogenesis. So various phases are there. The multiplication phase, growth phase, and maturation phases are there, through which the male and female gametes are produced. And we will move to the second step or second process, that is the sperm and egg recognition and the binding. So this binding is a biological process, and the sperms have few receptors uh, on the egg, and the egg also have some receptors for the sperm on their surfaces. So the zona pellucida that was involved in the sperm egg binding in the uh, egg. So the zona pellucida glycoproteins, which are found in the unfertilized eggs, contain a receptor called sperm, and that is receptor is modified after the fertilization. So the membranes of the egg, unfertilized egg, have the zona pellucida glycoproteins, and these glycoproteins have a specific receptor for the sperm. But these receptors are species specific, means if the sperm of the same species is there, then it will recognize. Otherwise, it will not recognize the sperms. Likewise, the egg receptor on the sperm. So, Golgi enzymes known as the glycosyl transferase. This enzyme catalyzes the addition of glycosyl residues from a donor nucleotide sugar, uh, UTP galactoside, to the terminal end of the O-linked oligosaccharides. So this enzyme catalyzes a few changes in the uh, sugars residues sugars residues on the sperm. So as in all enzymatic reactions, so these changes will make the uh, two gametes to recognize each other, and they will bind. So what are the acrosome reactions? So it is also an important process. So as a result of these the above changes, um, after the recognitions, there are few irreversible changes and binding is there. So as a result of irreversible binding of the sperm to the egg, the zona pellucida triggers the acrosome reaction. Okay. So this irreversible binding causes the acrosome reactions. So what happens in the acrosome reactions? So the outer plasma membrane of the acrosome fuses at multiple sites with the plasma membrane of the egg. Okay, so the fusion of the plasma membrane is there. So two of the important components are acrosine, a serine protease, and acyl glucosamine-based. So acrosine, what acrosine does, that it bores a hole in the zona pellucida. After uh, creation of the holes in the zona pellucida, the sperms can reach easily to the egg cell. So this is the acrosome reaction, and in the next lectures we will discuss the third step, that is the sperm and egg fusion, and then the after the fusion, the activation of the fertilized egg and the zygote will be discussed. So, hope you are satisfied with this detailed session. And if you have any query, please write in the comment box. I will resolve your query. This session is powered by Digital Version 2, Jyoti Vidhyapeeth University, Jaipur. Thank you.